Welcome to this explainer. Today, we're exploring the absolutely fascinating deterministic continuum that completely bridged the gap between a lifeless, rocky Earth and the very first living cells. We often tend to think of biology and chemistry as these two totally isolated worlds, but we're going to dive into the exact mechanisms that blur that line entirely. So, how exactly did inorganic rock and water spark living cells? Let's dig into this ultimate prebiotic puzzle and uncover how our ancient Earth basically acted as a giant planetary-scale chemical kitchen. As you'll see, life wasn't just some sudden magical event. It was actually a direct, inevitable result of geochemistry and thermodynamics. Here is our roadmap for this explainer. First, setting the Hadean stage. Second, the thermodynamic spark. Third, the concentration problem. Fourth, rock walls to lipid membranes. And finally, the first free-living cells. Let's get right to it. Section 1. Setting the Hadean stage. To really grasp the ingredients we had to work with, we just have to look back at the geological timeline of our young planet. And honestly, what's mind-blowing here is the sheer speed of it all. Earth forms about 4.5 billion years ago, and within just 100 million years, by 4.4 billion years ago, the first oceans condense. Remarkably, by 3.8 billion years ago, we already have carbon isotope data showing evidence for biological CO2 fixation. By 3.5 billion years ago, stromolites, which are preserved microbial mats, were widespread. Life literally did not dawdle. The second liquid water hit the scene, the chemical gears started turning. The big takeaway here? There is no hard, definitive line between chemistry and biology. If you look at the origins of life as a true evolutionary process, it implies a continuum of increasingly lifelike entities. The laws of physics and sheer gradualism simply drove chemical evolution forward. The Earth provided the raw, inorganic molecules, and through immense time and energy, these molecules complexed into the biological systems we know today. Moving on to Section 2, the thermodynamic spark. Early Earth needed a massive jolt of energy to spark these reactions. Mostly because life is fundamentally a non-equilibrium process. Now, what does that actually mean? It means life requires a continuous, non-stop flow of energy to survive, grow, and build complex structures, a process called dissipative structuring. Basically, if a system reaches equilibrium, it's dead. Done. At the origin of life, our planet provided immense thermodynamic potentials to keep things moving. We're talking everything from the constant bombardment of solar UV photons on the surface to wildly powerful chemical and thermal gradients deep under the sea. You can think of early Earth as having these massive, distinct chemical reactors cooking up biological precursors. On one hand, deep-sea hydrothermal vents provided geothermal heat, highly alkaline pH gradients, and intense redox chemistry, all in pitch darkness. On the other hand, you had shallow subaqueous basins providing UV light, dynamic wet-dry cycling, and interactions with atmospheric cyanide. Both of these extreme environments offered the continuous dissipation of energy needed to drive the synthesis of critical things like nucleotides, amino acids, and lipids. Deep-sea hydrothermal vents beautifully harness those temperature and pH gradients to literally pull electrons from chemical species like hydrogen and hydrogen sulfide. This flow of electrons reduces CO2 into vital organic precursors like formate and methanol. Basically, the inorganic hydrothermal vent itself acted as a massive catalyst. It utilized that thermodynamic driving force to push extremely difficult carbon-fixing reactions forward, which absolutely laid the groundwork for autotrophic life. Section 3. The concentration problem. But here's a massive hurdle for early chemistry. Once two molecules actually manage to react on a surface, how do they not just immediately diffuse into the deep ocean, never to meet again? The Hadean Ocean was huge, potentially up to 10 kilometers deep in places, with massive violent tides. If life was just a loose prebiotic soup, the vital ingredients would be way too diluted to ever form something as complex as a self-replicating RNA molecule. I mean, you can't just leave a bowl of chemical soup out in the rain and expect it to spontaneously organize, right? It demands concentration. Well, the answer lies in physical boundaries. Specifically, natural 3D rocky compartments made of iron and nickel sulfides found at hydrothermal seepage sites. These fossilized formations are essentially microscopic sponge-like labyrinths, and they acted as the very first rigid cell walls. Let's trace how this worked step by step. First, hot,
Reduced alkaline fluids seeped out of the crust and mixed with the much cooler, acidic, iron-bearing Hadean ocean water. This abrupt mixing caused iron sulfide to precipitate, naturally forming those porous 3D structures we just mentioned. The prebiotic syntheses occurred right on the inner surfaces of those metal sulfide walls. Because it was a confined physical compartment, the reacted products were completely restrained from diffusing away into the vast ocean. By trapping these organic molecules in microscopic rocky pores, the vents perfectly solved the concentration problem. This allowed those first self-contained redox reactions to really gather steam and complexify.
Section 4. Rock walls to lipid membranes. So eventually, life has to leave the rock, but we face a really severe biochemical puzzle here. The two oldest domains of life, archaeobacteria and eubacteria, use completely entirely unrelated chemical pathways to build their lipid membranes. Archaeobacteria use isoprenoids connected by ether bonds to a very specific glycerol backbone. Eubacteria, they use fatty acids connected by ester bonds to a completely different glycerol stereoisomer. The biological pathways to build these two membranes literally could not be more different. And why does this matter? Because it means their shared universal common ancestor had no biological membrane at all. Its membrane was that inorganic iron monosulfide rock. But if we look past those wildly different lipid boundaries and peer into their central metabolism, like mapping out the sequence similarity of glycolytic enzymes, we uncover a fascinating shared history. While some pathways definitely differ, archaeobacteria and eubacteria share incredibly deep, localized biochemical inheritances, especially when it comes to processing genetic information and core carbon metabolisms. They share the universal genetic code, ribosomes, and DNA polymerases. These are highly complex biological tools still beating inside your own cells right now, inherited from an ancestor that literally lived inside a rock. Researchers William Martin and Michael J. Russell hit the nail on the head with this. They point out that this immense evolutionary divergence happened while life was still physically stuck inside its inorganic rock incubator. The ancestors of archaeobacteria and eubacteria differentiated within the vent system itself, eventually leading to two entirely separate independent escapes into the open ocean once they figured out how to build their own lipid bags. Which brings us to our final section, Section 5, the first free-living cells. Let's look at what life actually packed for this grand escape when these distinct chemical systems finally broke free. Before breaking out of the vents, the last universal common ancestor wasn't just some primitive, simple droplet. It had already mastered insanely complex machinery inside its rocky home. It possessed the universal genetic code, ribosomes and tRNA to build proteins, DNA and RNA polymerases for replication, a robust core carbon metabolism, and F1, F0 type ATPases to use as energy currency. All of this evolved while life was physically anchored to the hydrothermal vent, completely relying on those constant geochemical gradients to survive. Once they independently evolved the ability to synthesize their own distinct lipid membranes, swapping out rigid rock walls for flexible biological ones, they could finally maintain their own internal chemical gradients. And boom, over 3.8 billion years ago, the first true, free-living cells were born. They detached from the hydrothermal vents, forever leaving their rocky origins behind to conquer the vast Hadean ocean, and eventually the entire planet. This whole journey from geochemistry to biology leaves us with a profound, almost mind-bending realization. Life wasn't a freak accident. It was the result of a steady, deterministic continuum driven by temperature gradients, rock pores, and basic thermodynamics. If the prebiotic puzzle was solved by universal physics and chemistry here on Earth, this exact continuum is likely playing out in hydrothermal vents across the cosmos right now. So the next time you look up at the stars, ask yourself, how many other rocky, watery planets are currently acting as a giant chemical kitchen? Thank you so much for joining me for this explainer.